Hey guys, this is Tiffany Martin, and this is Medical Microbiology, and we are starting. This is the first lecture series in the Chapter 9, which is Controlling Microbial Growth in the Environment. And my lighting's not very good here, sorry about that. And so we are learned a lot about this now that we've done the COVID-19 and everybody is trying to disinfect their environment. So let's kind of figure out what we know, what we don't know, and what we can do about it. So let's start with the PowerPoint, and this one we actually did cover before spring break, but who remembers that? That's been so long ago. So let's start back at the beginning and work through the PowerPoint, and I'll be going back and forth a little bit between the two. Okay, so first thing we need to do is we need to discuss some terminology. I need to make sure that you know the actual definitions to words that we get thrown out a lot in the media and in just regular life, and let's see if we know the medical terms for that. So reviewing those, we have, let's see, I can go that, Chapter 9. We need to talk about sterilization. Sterilization is the removal or destruction of all microbes, including viruses, including endospores. And removing endospores, remember that's where it layers different layers on top of that, right? Endospores are where it forms the cell, lays down layers after layers after layers, and you get this spore that can survive in extreme conditions. Oxygen for sure, but other things too that might be extreme heats, it can survive a lot. So if we can get rid of endospores, that's the definition of sterilization. Boiling water does not cut it. That won't sterilize. So when we use terms like, oh yeah, put your toothbrush in the microwave in some boiling water and let it boil it so it'll sterilize it, we're not really sterilizing it. We are disinfecting it. We're getting rid of a lot. It's a good thing to do, but we're not sterilizing it. Same thing with baby bottles. Throwing those in boiling water. It's not going to sterilize. So to get sterile, we need to remove everything, including endospores. And that's going to require some heavy-duty methods, and we'll talk about what methods that requires. All right, so sterilization, we know what that is. What else do we need to know? We need to talk about disinfectants and antiseptics, disinfection and antisepsis. Antisepsis is the process where we remove pathogens. I'm skipping ahead. Disinfection and antisepsis, that's on the next PowerPoint slide, disinfectants and antiseptics. Antisepsis is the process of removing pathogens from skin or living tissue. Antiseptics are the chemicals that we use in antisepsis, okay? So antiseptics and antisepsis go together. Disinfectants, we know what that is. Those are chemicals, or it could either be methods that we remove pathogens from inanimate objects. So we disinfect our countertop. We use antiseptics when we are trying to clean a wound or living tissue. So make sure you understand the difference between disinfection and antisepsis. That all has to do with removing pathogens. The difference is on what type of surface you're doing it. Is it living tissue or is it an inanimate object? So that's the difference there. Now there is another term called aseptic. That's not the same thing as antisepsis or antiseptic. Aseptic is the term we use when we describe an environment or a procedure. So this is a place or a process, environment or procedure that is free of contaminations by pathogens. So we use that word when we talk about aseptic technique. We talked a lot about that in lab this semester. When we flame our loop, we're using aseptic technique. When we stop, when we are looking at our plates and we take the lid off very carefully and kind of leave it protecting it from that. That's aseptic technique. We're trying to avoid contamination from things falling on that. When we hold things at an angle, when we're dipping and swishing, we've got our tube, we've got our loop, and we hold that tube at an angle when we dip and swish, and that's to keep things from falling into it. If we're there, we're not doing this. All of that was aseptic technique. We are creating an environment, or we're doing a procedure that's going to help us reduce contamination. Now, all of this, disinfection, antiseptics, disinfectants, aseptic technique, antisepsis, all of those things, none of that is sterile. we're reducing the number of microorganisms. We are not removing them completely. So none of that will sterilize. So make sure you are aware of that. Okay, so terminology, still working through that. We covered disinfection and antisepsis. Degerming, this is the scrubbing. This is when you remove microbes from a surface by scrubbing. And this scrubbing action, when we're talking about washing our hands or if we're talking about preparing an injection, this scrubbing action is just as important as the chemical we're using. That term is called degerming. So, and that doesn't get used a lot. All right, so make sure you know what that is. All right, what else do we got? Other terminology, sanitization. This is not sterilization. It's not even disinfection. Now we're going down a level from that. Disinfection will remove pathogens. Sanitization removes some microorganisms, but it may leave pathogens. But it removes them to a level that is considered acceptable public health standards. So you'll see sanitization like on your silverware at a restaurant. It's in a little I don't know, a little package or something. It says sanitize for your protection. The dishwasher sanitizes, that kind of thing. It removes most pathogens. There may still be something there, but the average person is able to, their immune system is normal, typical, and they're able to fight it off. So if it's sanitized, it's definitely reduced the number of pathogens, but it doesn't remove them completely. But that's okay. When do you need to be worried about that? Well, if you don't have a typical immune system, it may not be enough for you. If you're going through chemotherapy and they're knocking out your immune system, they tell you don't eat at restaurants, limit your exposure to the public. Sanitized may not be enough for you if you're immunocompromised. So that's when you need to be careful. But for regular people, sanitized is good enough. What else do we need to know? Pasteurization. This is another term that we use when we're talking about reducing the number of microorganisms. This also is not sterile. This reduces the number of organisms, and we use pasteurization when we're talking about food and beverages. We'll use pasteurization with our milks, with our orange juices, things like that. What we're doing is we are heating up our milk to get rid of a lot of microorganisms, won't get rid of them all, and then we cool it back down. We have to balance. We don't want to boil the milk. It changes the taste. It changes the protein structure and changes the taste. So we're going to heat it hot enough to kind of get rid of a good many things, but not so hot that it's going to change the taste. So that is what we're talking about when we're pasteurizing milk. Now you'll know that if you leave the milk in your refrigerator, and you don't touch it, even if you don't open it to the environment where it could be contaminated, after a while, even in the refrigerator, it's going to grow things. That's because there was microorganisms still in there, but they are at an acceptable level. The milk is not spoiled. If you drink them, even if you're drinking microorganisms, your immune system takes care of them. It's not a big deal. So that would be an acceptable level. So that's pasteurization. Okay, let's see, terminology. What else do we need to know? Okay, so we did this in class. This is an example of some of those critical thinking questions that comes up on tests. You need to know some things, and then you need to think it through and think about it. So I'll kind of go through this. Again, we're not together, so you can stop this video. You can look at it on your own. You can dial up your lab partners. You can get with them on different virtual hangouts and try to go through these. It will definitely help you if you try to work it out versus just listening to me talk to you about it. But these are some of these examples of critical thinking type questions. Okay, so this is going to ask you whether a disinfection method or a way to reduce microbial contamination is germicidal or germostatic. Germicide is when you kill something. Suicide, germicide, you're killing something. Germostatic means you just stop it from growing. If conditions return to good conditions for growth, if it's only germostatic in nature, the microorganism will start growing again when environmental conditions are better for growth. So you're going to perform this experiment, and then you want to determine whether this antimicrobial that you're going to add is germicidal or germostatic. We're going to be talking about bacteria, so you could be a little bit more specific and talk about bactericidal or bacteriostatic. Bactericide kills bacteria. Fungicide kills fungus. Viricide kills viruses. Germicide is kind of an a big umbrella term. It's talking about all of them. Okay, so what does it say? We're going to inoculate E. coli in 2 test tubes. I actually have this kind of written out. All right, I've tried the whiteboard on the computer, and my drawing skills are not very good, so I'm just going to kind of hold this up and see if that helps you. But how does this work? So we're going to start with 2 sterile tubes. It's got broth in here, sterile media. I'm going to inoculate both of them with E. coli. So dip and swish, add a little bit of E. coli to both tubes. One of them, not that one, this one, I'm going to add a drop of an antimicrobial. So this is a chemical that is going to inhibit the growth of bacteria. My question is it going to kill this E. coli, or is it just going to stop it from growing? All right, so 1 drop of antimicrobial, this is my control tube on the other side, no antimicrobial here. I let it go for 24 hours. After 24 hours, I get growth in my control. I get growth in my control tube, so you can see I got that, which we would expect. E. coli is going to grow. The one that I added an antimicrobial to, I didn't see growth. So it definitely stopped the growth. Did it kill the E. coli, or did it just inhibit the growth for the moment in time? And when environmental conditions get better, is it going to grow again? All right, so work through your groups and see what type of process would you use to go through that, okay? All right, so I'm going to go forward in time. I'm going to pretend like you stopped this video. And the best way to do this is we know that I've got antimicrobial in this environment and it's not growing. But I want to know, is there bacteria here that is capable of growing? So if I take this and I inoculate a plate or a tube, could be either. So we'll do a plate. If I inoculate a plate, with this, if I get growth on that plate or that tube, then I know I didn't kill it. Then I know that this dosage of antimicrobial was bacteria static in nature. It inhibited the growth while that concentration was high, but when I diluted that concentration of the antimicrobial and put it on fresh media, either a plate or a broth, I got growth. So now I know it's bacteria static in nature. If I had inoculated it onto a fresh media and I got no growth, then that tells you even when environmental conditions are good, then it was bactericidal in nature. Because of this dosage of antimicrobial, it was enough to kill any E. coli. And when I put it on fresh media, so I diluted the antimicrobial and put this bacteria on fresh media, there wasn't enough to grow. It was dead and wouldn't grow. That's how I determine whether it's bactericidal or bacteriostatic. Okay, I hope that helps. I hope you're able to work through that a little bit. All right, so what else do we need to talk about and go? And make sure you work through some of those questions in the back of the book before the test. This is how you really know that you're understanding it, not just memorizing things. All right, what else do we need to know? We need to know about microbial death rates. When I spray it with some disinfectant, I don't kill everything immediately. There is a time factor. And if you're looking at your disinfectants and you're looking at the instructions, it'll say allow it to remain wet for 2 minutes or 4 minutes. Or if you leave it for 2 minutes, it kills this. If you leave it for 4 minutes, it kills this. And what's that happening is that for every unit of time, you get about 90% of microbial death. So 90% of the microorganisms in the environment will die for every unit of time, which sounds pretty good. 90% sounds like a lot, and it is a lot. But it also, you have to remember that microorganisms, we're talking about some really large numbers. So if I start with 10 to the 9th, number of microorganisms in my environment, if I kill 90%, I still have 10 to the 8th number of microorganisms, which is a lot of microorganisms. If I start with 1 billion and I kill 90%, I'm still left with 100 million. That is a lot of microorganisms. So allowing this time factor and killing 90% each time will eventually get you down to germicidal, or you have killed everything. If you stop and you don't leave it wet for long enough, it doesn't stay in contact with microorganisms, you could stop before you get to that 0. Then the ones that did live, they'll be able to replicate and go again. So do keep this in mind. When we're talking about microbial death, we are referring to permanent loss of reproductive ability. So we have permanently stopped all the microorganisms from dying when we get to that point in time. All right, so keep that in mind when you're looking at your disinfection techniques and making sure you get the job done. All right, what else do we need to know? We do need to know how they act. We're going to go through a lot of different agents and ways to inhibit microbial growth, and they act in different ways. And so it's always helpful to know how they're acting so we can determine what our risk is to ourselves and to our families. Two big categories. One is alteration of cell walls and membranes. If it acts on bacteria cell walls, it's not going to damage us. That's good. We don't have a cell wall. If it acts on membranes, it could be toxic to humans as well. Anything that alters cell walls and cell membranes is going to weaken those cells, and then just water kills them. This will kill alteration of cell walls and membranes. If we remember an enveloped virus, the outside of that envelope is just a cell membrane, right? It's the previous host cell membrane. SARS-CoV-2 virus that we're dealing with now is an enveloped virus, which means it's actually one of the easiest things to kill. If we can disrupt that envelope, then it's not going to infect us. And so even the mildest disinfection techniques, disrupting that cell membrane will take care of that. So that's good to know. It's very easy to get rid of. Another category of these agents are agents that damage proteins and nucleic acids. Anytime you mess up proteins, anytime you mutate DNA, you're going to cause problems. This also may cause side effects in humans because we have proteins, we have nucleic acids. We have to be careful about that. So knowing how microbial agents work is beneficial in how we protect ourselves. Okay, this is another critical thinking question. We did this one in class as well when we did this. I'll kind of go through it and you can kind of, actually I'm going to leave this one for you. What I want you to point out when you're reading these questions, let's read it carefully. This is a question that says, would you expect a gram-negative bacteria or a gram-positive bacteria to be more susceptible to antimicrobial chemicals that act against cell walls? And then it says, this is a test question, so your answer should be based solely upon the nature of cell walls. Now we know the environment is sometimes different and there's multiple things that come into play, but for a test type question, they're saying base this only on the nature of cell walls. All right, so what do you need to do to figure out this question? Well, first thing you need to know what a gram-negative bacteria is and a gram-positive bacteria is. So you have to know that in your mind. Okay, gram-positive, cell membrane, and then thick layer peptidoglycan. Gram-negative, cell membrane, thin layer peptidoglycan cell wall, and then an outer membrane. This is a chemical that only acts against cell walls. So presumably, based on this question, it's not going to interfere with the outer membrane. It's not going to interfere with the cell membrane. It's only going to damage cell walls. So which bacteria is more susceptible? Well, in this case, gram-positive bacteria is more susceptible because the cell wall will be directly in contact with the chemical. The gram-negative bacteria has that outer membrane that's going to provide some protection. So in this case, a gram-positive bacteria is more susceptible. So work through some of those questions and see what you can do. Okay, all right, so we will stop this video and pick up with the next one and talk about what types of decisions we have to make when we're trying to figure out what type of microbial control methods we'll use. So we'll stop here and pick up on the next video. Good? All right, keep going. Thanks, guys. Bye.